pop into screen right now. There we go. Just popping into screen,
there
everybody.
We go. Crawling
We're
my way into your television set.
That's right. We're crawling in here. The TWIS team. We crawled in here today. We made it. Got here finally to talk about science. Y'all ready for a show? Hey, Aaron Lore. You think so?
I think so.
I
think I might be ready.
Yeah, get it ready. All right, everybody. It's time for us to start this show we call This Wing in Science.
This Wing in Science.
Caw-caw! Caw-caw! Everybody be your favorite bird. Caw-caw!
My favorite bird call, it's the begging of baby crows.
That's good.
Wow.
Actually, that's more the sound of the baby birds as they're eating the food.
Yeah. I gotta pay more
Baby
attention
crows are
when
more
I'm out
like,
in nature.
And then the scrub jays are like, right, right, and they screechy like that.
I ran into an egret the other day that went blah 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 blah.
I like that one too.
Yeah, it was really good.
Okay, I need to find some egrets and listen to the egrets.
Blah blah blah. It's so good.
It's fantastic. Everyone, record your favorite bird song for us. And send us your favorite recordings of yourself impersonating your favorite bird song, because that's fun. Well, here we are in another episode. I hope we all sound fine. Are we 5 by 5? Are we allowed to say that? 5 by 1?
5 by
1 by
I'll
5.
settle for 5 by 4.
10 by
5
10.
by 4?
Yeah.
4 by 4.
Suddenly we are off-roading in the world of science. Science off-road. Oh, I see where this show is going already. Everyone, Blair has warned us that California has power grid problems.
I don't remember what the exact words are. It was like a power flex or something where, like, basically,
it's called rolling blackouts.
It's too darn hot, and they expect everyone to run their AC at the same time, and they don't think all the grids will make it. So
they're not gonna make it, Captain.
I think I'll be fine in Redwood City. I mean, I don't have AC, so and it's not that hot right here. But it was 90 something at my office today. So
let's talk about science.
We'll see. I think it'll be fine.
I think I just want to imagine like the lead engineer at PG&E having like the Scottish accent from the engineer from Star Trek. We've got to have more power. And then he's like, redistributing power to engine number 5, Captain. Yeah. Anyway.
Divert power from shields. What could go wrong?
Diverting power from San Francisco. Yes. So, Blair and Justin may or may not make it all the way through the show, but it's great so far. And you're wearing your Trek shirt. It's
I'm ready for everything.
I think we're just doing the after show pre-show.
Yeah.
Shields up, everyone.
Shields up.
All right, time to check. I don't know what you're going to check. I'm just going to start.
Am I checking something?
Yeah. My computer's on.
Check your tricorders, everyone. Are we ready to go?
This can't be right.
Hi, Justin.
This is funny. Jess is the one who's now exasperated by these two.
No, it's always the case. That's just this, ladies and gentlemen. I apologize for the sometimes rambling, periodically unprofessional behavior of my co-hosts. Let's get right to the
actually started
science.
Recording yet?
Oh, how come we're? Oh, but we're live.
Oh, we are live. Yes.
Yeah,
I mean,
we've been
this
live
is
for 5
recording,
minutes.
but like,
Well, What else are you talking about? What other show are you doing? Ladies and gentlemen, there's apparently been something put into the waters of both San Francisco and Portland. Well, reports are just now coming in of the effects of this unknown substance.
Oh no!
It's obviously clear nobody should be driving or operating heavy machinery until scientists can figure out what the heck is going on.
Oh, good Lord.
Laugh till you cry, people. Laugh till you cry. All right. It is hot down here. I know it's not San Francisco and Portland, but yeah, it's time for the show. Are we ready to do this?
Yes.
Here we go. We're going to do this. We are going to do this beginning in a count of 3, 2. This is TWIS, This Week in Science, episode number 835, recorded on Wednesday, July 28th, 2021. Who wants to CRISPR mosquitoes? Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with mosquitoes, Neanderthals, and pterosaurs. But first...
Disclaimer, disclaimer, disclaimer. For some, life is a brilliant chance to explore the unknown, to take on the challenges of collecting data, forging an understanding of how things work, and in doing so, seeing the world without the veil of human bias. But for most people, information is rooted in what they believe, not what has been rigorously confirmed. A Yale University poll conducted late last year shows that the majority of Americans believe global warming is happening, 72%. We'll ignore the other 28% for now. Actually, let's just ignore them completely. It's not the only thing they're going to be ignorant about, and it's only a matter of dreadful moments before they segue into talking about the other ignorant beliefs they are metastasized with. More interestingly, is that while not 72% believe global warming is real, only 55% believe that most scientists think global warming is happening, which is an astonishing reveal. It means that 17% or more of Americans who believe global warming is happening think it's the scientists that still need convincing. If there was only some way to reach them. Further, it found that only 57% think global warming is caused by human activity, which means then 15% of Americans could think that global warming is happening, that scientists haven't figured it out yet, and that it's just a natural phenomenon. Information, assumptions, and conclusions based solely on what they believe to be true. And it's an important note here that while climate change is often framed as a political issue, it isn't one. It just seems like it, because 100% of elected officials in the U.S. who deny climate are Republicans. And while they are only a slim majority in that party, they are the majority in power and hold tremendous influence on what many people believe. While all of these numbers are the way they are because maybe it's the 25% of Americans who reported only hearing something about global warming once a week, what all this suggests is that to get real climate action, all we need to do is convince the media to cover it so that more scientists will believe in it. Because once scientists are on board with something, the next thing that happens is This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about science. every time I say that, I'm like, I'm going to say something else. And no, it's science, because that's what we talk about. This past week in science, what happened? A lot of things, and we're going to talk about them. I have got the mosquitoes. I brought the mosquitoes to the show this week. I apologize, but I think some people might appreciate this story. Other people might not. We may talk about it a little. Additionally, I've got some more folding and some human genome news, as well as a story toward the end related to baby mouse dreams, because those are interesting. What do you have, Justin?
I've got Neanderthal blood. Bees
Of
on
course.
caffeine. Bees on caffeine. Oral biome history. And some really, really bad news.
Oh, are we going to just do that first?
No, I have a whole thing where I'm going to end my portion of the show, at least, with the worst possible news. And then we'll go off to mouse baby dreams.
Okay, we will go off to I like that. We will go off to mouse baby dreams at the end. All right, Blair, what is in the animal corner?
Well, I have a story about dogs that know you're lying. I also have baboons and fitbits and baby pterosaurs.
Oh, we've got babies on the show today. Yes, we brought the babies. If all of you out there are listening with your babies and you want to make sure that they hear the show every week, know that you can subscribe to This Week in Science. We are a radio show, but we are also a podcast. You can find us all places podcasts are found, and you can find us on YouTube, Facebook, and Twitch. We are also TWIS Science on Instagram and Twitter. Our website is twis.org. All right, now let's dig into the science. I'm just gonna go straight to that pointy little proboscis, and we're gonna talk about mosquitoes. We'll just start right there. With the reality that scientists are working very hard to try to get rid of them. They are. And we have talked before on the show about the idea, the new technology, the evolving technology, which has made its way to the forefront in not so much in vaccine research, but also in animal research. It's called gene drive. How can we put a gene into a species that takes over and doesn't let other variants of that gene exist anymore? So that in the case of mosquitoes, you put a certain gene into the mosquitoes and they can no longer bite their prey because it changes the shape of their face, basically changes the shape of their face so they can't bite their prey to transmit the parasite that causes malaria and so many other viruses and diseases. Additionally, they can CRISPR in a gene to make it so that females can't lay eggs anymore. By doing this, if the gene takes over, suddenly you have an entire population of mosquitoes who can no longer bite their prey or lay eggs. And what is the outcome of this kind of a population? Death. They die, not us. The mosquitoes die.
Well,
So the goal is to reduce
maybe.
mosquito populations and reduce disease in humans.
Yeah.
But what was your point, Justin? What do you want to say?
Well, I was, if there was one thing that I would just remove from the planet without any sort of regret, it's always been
people.
mosquito.
Yeah.
However, I then learned that there are major pollinators. And now I think it's a really, 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 really bad idea.
They are major pollinators and they are a major food source for other species. So if we get rid of them, what happens then? So with the gene drive, you hope that something like this will stay within a particular species and that it won't jump between species.
That was what I was going to ask about.
Yeah.
That'd
So
be
far,
less
we don't
scary.
know the answer to this question, but the study that is out this week in Nature has achieved gene drive in populations of mosquitoes in a lab to kind of virtually real scenario. So we're not talking about mosquitoes in little tiny aquariums. They have built climactic chambers that are supposed to have more realistic environmental aspects to them. And this is part of the process of testing gene drive. The question is, you can make these things happen in the lab, and you might get to 99% suppression of other gene variants and 99% effectiveness of the gene that you've stuck in there, that's driving its way through the population, making its way through 99% of the population in the laboratory. But what happens out in the wild? What about the individuals that get eaten? What about the individuals that just die? What about the individuals that just can't handle the cold? How do their genes, do things change when you put them out in the wild? And so they stuck them in some very large feeding cages and showed that, lo and behold, when they put the gene drive in these populations, it still worked. So laboratory to realistic-ish situation, it works. Gene drive still continues.
So the whole idea of gene drive jumping from mosquito to anything else seems to me implausible. Like, not just like it might not happen, but like I don't even know how you could create that to make it do that. Like that, like to even engineer something that you would want to do it would seem impossible. So I don't think that's a fear. But you're like, why like you're saying, all the things that feed off of mosquitoes.
Right.
Bats,
they didn't have any of that in there. There were no bats.
No.
No bats in these cages. Yeah.
But then it's sort of, if we can do this, why can't we give them like the butterfly proboscis, something when they can change their diet and eat something else?
Right.
Transmorgify them into a creature of our own design.
Just selective breed them, basically. Just make them a much less dangerous version.
Yeah. I mean, I think if we could, these are the bioethical conversations that need to take place. Which, yes, this seems to be working. But what happens when it does actually get into the field? What exactly does that mean? Knowing that this bridge situation with the large field-like cages for this paradigm of studying gene drive, not in the field, but close to it. And then we're going to learn more about how the gene drive works in this situation. I mean, that's good. It's the ethological way to study this, but we still have to have these questions of, is this the kind of gene that we want to change population structures? Is there some other gene that we should be working on? Should we just be affecting feeding behavior and not ability to produce offspring? Should we, are there better technologies?
Right. So, and the real ethical one, way to go would be to alter our own genes so that we were not appeasing a meal. That would be the only ethical way to do it. Is as a species we would decide to change ourselves, deciding to make this whole change for environment and altering another species. It sounds again, until I found out that they were pollinators, I didn't care. Some bats went hungry, pick another bug. There's other bugs out there. You didn't get them all. But now I'm worried, like, how we don't necessarily, or we definitely don't have an idea of how important they are to our biome environment overall.
Yeah, I think the big question for me is we've seen a lot of gene flow between species of lots of different kinds of species, but mostly plants. But it does happen in insect species as well. And if species, we think of them in these little boxes, but really there's this continuum of one species into another, and with subspecies in between. So if you have subspecies interbreeding, that could lead to this kind of a gene driving through various populations and getting into different species, then that's the big question. Will releasing something like this into the wild have an effect we can't take back later? And we really need to answer that question. But
and I think
we're
it's
trying
still
to do the experiments. But go ahead.
Yeah, and I think it's still a really important thing to explore because malaria is extremely dangerous. That's not the only thing that mosquitoes spread too. And we are already messing with ecosystems due to climate change. So as much as I want to keep things how they were, mosquitoes are now going where they weren't historically because of climate change. So even though I want to keep pollinators, and that's an extremely important thing to keep in mind, you also have to recognize that like this is kind of a runaway cart with all sorts of things. And so maybe the spread of disease through mosquitoes in places where they're not supposed to be because of climate change could be worse than potentially losing the pollinators themselves.
Yep.
So there's yeah, it's complicated. I'm just saying it's I know I'm usually the one saying like keep everything the same, but this is one of those situations where I recognize it is extremely complicated and this research needs to be done.
Yeah. Yeah, I don't think we should stop the research. I definitely think it should continue, but I think it needs to be done with continued conversations with communities, and we need to have these conversations to see, what do we think about this? What do we want to do? What's more important?
Very interestingly, I'm getting word from the science line here. There's a Danish malaria vaccination on its way, where the antibody is transferred to the mosquito when they drink the blood, and they have found out that it can kill the parasite in the mosquito. So now you have
That's impressive.
Now you have a vaccine that prevents you from getting sick, but also is curative to the mosquito. That sounds like a better strategy. You might want to focus on that one.
I'd like you to focus on Neanderthals right now.
Okay, so...
And their blood.
Yeah, Neanderthal blood. So, there's not a whole lot of samples out there, right? Before genetics was a thing, one of the ways in which anthropologists could try to determine the origin or migration of human populations was through blood typing. When genetic data became available on humans, sort of blood typing became largely relegated to making sure that you got a successful transfusion. Now the two techniques are being combined in a way to sort of look back into the history of Neanderthal and Denisovan evolution, health, and some of their vulnerabilities based on their blood work. Now, how do you get blood samples from a 40,000-year-plus old Neanderthal?
Ask nicely.
Ask nicely.
Give him a lollipop.
I don't know. You have to, I don't know.
I have to
thaw
tell you?
it, put it in the microwave? What?
Aha! So they haven't. So in these fossils, and there has been we could get some recognition that there was heme and some very old bones and stuff, but no, they didn't get a blood sample. What they did was they got enough of the genome decoded that they can look at the genes that code for making the blood, and within that they can see the alleles, they can see the types, they can see the blood through just the genome. This is Sylvana Condemi of the Centre National de la Recherche Scientifique and her colleagues in France investigated the previously sequenced genomes of 1 Denisovan and 3 Neanderthal individuals ranging from 100,000 to 40,000 years old, and managed to determine blood types and analyze the implications of what was there. So there are some, I guess there's more than I thought. There's it says here 43 different systems for assigning these blood types. And they looked at the sort of most common types first. And they found that Neanderthals and Denisovans are polymorphic for ABO, which is like current modern humans have A and B and O blood. That's not so different. And actually they shared, these blood groups also shared alleles that are recurrent in modern sub-Saharan populations, therefore making it consistent that the idea that Neanderthals and Denisovans have African origin. something that has not been hugely argued against, but there have been some indications or some talk that maybe Denisovans could possibly have a non-African origin, or at least not as recent. But it looks like, yep, they're consistent with sub-Saharan populations in terms of blood typing. In addition, a distinct genetic link between Neanderthal blood types and the blood types of an Aboriginal Australian and indigenous Papuan suggests the possibility of mating between Neanderthals and modern humans before the modern humans migrated to the Southeast Asia area. Neanderthal individuals also had blood type alleles associated with increased vulnerability to diseases affecting fetus and newborns, as well as reduced variability of many alleles compared to modern humans. The pattern, they say, is in line with existing evidence that we've also found from other things that links low genetic diversity and low reproductive success with the potential demise of the Neanderthal populations. They also found genes in both Neanderthal and the Denisovan associated with strong resistance to norovirus, which can cause upset stomach. And also just goes to show these viruses have been everywhere, and we've been dealing with them forever. It's not new. Yeah. So it confirms African origin, some weaknesses in their fertility, and a little bit of virus fortitude amongst our ancestral cousins. So this is published in the Public Library of Science Online. So if you want, you can go check it out. That's free. Open to the public.
That's awesome.
Your local library of science.
And everyone who is on the YouTube channel right now, I apologize for our YouTube chat. We will I'm going to see what I can do to slow the chat, but I hate to have the people who are conscientious chatters shut out. So, Fada, if you can keep working on that, that would be great help. I appreciate what you're doing there. But let's continue, and remember that our chat rooms are safe spaces for our friends, and we're going to talk about science. Okay? So don't be a jerk. Don't use terrible language, and we don't accept that kind of behavior here. All right. As I'm saying it.
Yeah.
Yeah. All right, Blair.
Yes.
What was your next story?
It's just a little story about dogs. So dogs, they're pretty intuitive. You think, they might know a little bit about what's going on in your brain based on your body language and what they know about you. And so this is a story that I brought for the quick stories because it's an excellent headline, but I think I really wanted to just look at the experiment itself and talk about it for a second. So the headline is that dogs can tell when people are lying to them. So this is from the University of Vienna. And they claim that they can indeed tell if a human is being an unreliable deliverer of information, if they are lying, if they are being knowledgeably obtuse, you could say. So they did a study with hundreds of dogs, and they were taught to follow the advice of an unknown human in choosing which of two bowls contained a hidden treat. This is very similar to stories we've talked about before on the show, where they just kind of pointed a bowl, and that is the bowl with the treat in it. By following the advice, they get the treat that's in the bowl. Then they allowed the dogs to watch as another unknown human moved the treat from one bowl to another while the second unknown human watched. So then they were able to see that this human knew that it wasn't in the bowl that they were showing them. They also did one for control where the human didn't see where the treat got moved to. And then they came in and pointed at a bowl. And so then they conducted the same experiment. Which bowl will the dog go for? The dogs ignored the human advice if they weren't in the room. So they appeared to not care at all about what bowl they were pointing at because they weren't there. How could they know? So that's interesting. I think that's actually the interesting piece of this experiment is that they could identify what knowledge that human had. But what they claim is kind of the crux of this study is that when the human knew where the food was and pointed at the wrong bowl, half of dogs ignored them. So that to them means that the dogs knew the humans were lying. I would say that means the dog knows where the food was.
Well, either the dog knew where the food was, but I mean, 50%. That's like chance.
Yeah, exactly. That's my other problem.
That's random. That's like
yeah,
could be either way.
The other thing that's interesting about this is that they then compared it to similar research with humans under the age of 5, macaques, type of primate, and chimpanzees. And in those experiments, the children and the primates were more likely than dogs to follow advice of an quote-unquote obvious liar over what they knew to be true. So what they're saying is this suggests that the dogs were less trusting of unknown humans with advice. I also think that's kind of a leap. I would also like to know whether they used treats that smelled at all. I would like to know if they did this again with a trusted source, how they would act. I think that this is an interesting first step to recognize that they behave differently than other animals in previous experiments. I think that's really what's interesting.
That's interesting. Mm-hmm.
And I think it's interesting that they knew, like, you don't know anything. Let me figure this out for myself. I think that's really interesting. I don't know if this really points to dogs knowing when humans are lying, but I think it does mean that dogs know what humans know if they're there to see it. So it's almost like this is a reach, but it's almost like they can put themselves in the human shoes and be like, okay, I know what that person saw, so I know what information they might have. It almost implies object permanence, which I think is really interesting, but that's a whole nother thing. Anyway, dogs are smart. They're social. They understand how to communicate with humans, and they might be better at that than wild animals and babies.
Great. Might be. More study needed.
Yes.
For sure.
For sure.
Larger sample size.
Yeah.
Yeah. Let's not use treats. Let's use food. Let's use toys that don't smell.
Yeah.
Yes. Figure it out.
Or put them on the other side of a piece of glass or something. Yeah.
The smartest animal I ever met was a mule.
I believe
Not
that.
a crow, huh?
Not a crow. It's a mule. A lot of people think mules are stubborn. It turns out they're not actually stubborn. That, like, if they don't want to do something, they get that they're bigger than you, and so they just don't.
When training animals, we always talk about kind of like, this is an extra scientific way to talk about it, but basically a bell curve, right? So if they're really, really not bright, they're pretty hard to train because they can't associate reward with behavior. But there's this sweet spot in the middle where they're smart-ish, but not too smart to outsmart the training, right?
Right.
And so this is where, like, my dog was too smart to learn to ring the bell to go out to pee because she figured out I can ring the bell whenever I want and they'll take me outside. Too smart. So you want that middle. Not too smart, not smart enough.
You want a dog that is not as smart as your child.
Yeah.
Yeah,
because they don't speak English, so it makes it really hard to reason with them.
It does. Don't worry about reasoning with your dogs. No, that just goes down. That's bad paths there. Speaking of reason, artificial intelligence has a lot of it, potentially. Following up from last week's story about the Rosetta Fold open source protein folding predicting AI that was released. Basically at the same time, AlphaFold came out and made its source open to the public as well so that people could peek behind the hood and see how it worked. Additionally, they published that they were predicting the structure of more than 20,000 human proteins in addition to almost all the known proteins from 20 model organisms: E. coli, fruit flies, yeast, soy, rice. Lot of organisms, mice. They're going to add to this database that's going to be a publicly available, searchable database. It's a combined total of over 300,000 protein structure predictions The company has released all this online. It's going to be held by the European Bioinformatics Institute based at the European Molecular Biology Laboratory. And as of last week, researchers were going into this database and searching for their proteins. Biologists looking for the proteins that they study to see if the structure had been adequately predicted. Well, it's not that great. It increases us from our previous amount of 18% of the human genomes, or the human proteomes of the human, 18% of the human proteome, to about 40%. We now have pretty accurate structures, very accurate structures, for about 40% of the human proteome. Now that's a lot, but that leaves a lot of proteins still up for grabs. And while I was snooping around on Twitter last week, I saw a lot of researchers posting images of the proteins that had been, the structures that had been predicted for their proteins, and just shaking their heads and laughing and going, "This is telling me nothing. This is totally wrong." And so a lot of researchers are still going to be waiting for their proteins to work. And the reason behind a lot of this is because there are a lot of proteins that they only get into their final conformation when they interact with other molecules. So because we don't know what the other molecule is necessarily, or we don't have that information involved in the artificial intelligence, the deep learning that would predict all the structure, the end structure, there's no way to know what it would look like, because the structure is going to be determined by some other forces, molecular forces. But that said, opens up our catalog of protein structures massively, adds to this ability for researchers to be able to link together structure and function and understand a lot more about us and the world we live in. And it's just the beginning. It's just the beginning, which is so exciting. And then I think additionally a story that was, I think that's all I want to say about this. Never mind.
Okay.
That's all I want to say about this. I was thinking about all the things I wanted to say about it. But some of the other proteins that are not included in this catalog are also larger. So they're too big, and AlphaFold set a cutoff for the size of the protein, the number of amino acids that would be included in the proteins.
The bigger it is, the more crazy different ways it could fold. Also.
Yeah. So researchers working on the big giant proteins, you're out of luck, but there are fewer of you. So, yeah.
I know.
Whatever.
I picked the wrong protein to research.
It'll happen eventually. You'll get there.
Don't give up, science.
Don't give up. What do you have next, Justin?
Bees on caffeines.
Okay.
Yeah, so there's been past studies that showed that bees like caffeine. You can they will more frequently visit a caffeinated flower, or one that is sort of treated with some caffeine. And these researchers thought, okay, that's interesting. They seem to like caffeine, but maybe this is just this is something that's going on within the bee itself. Maybe they're showing up more to that flower because it's also more interesting, or they can smell it better. Maybe it's affecting the bee's memory of where that flower is.
Isn't caffeine addictive? So
No, why would you say that? There's a Starbucks on every other corner now, but that doesn't mean it's addictive. It just means it's really, really good.
So
you have to have it to start
So they're
your
just
day,
getting
otherwise
bees
your day is
hooked,
terrible.
is what's happening.
Well,
Hooked on caffeine. Well, caffeine is supposed to enhance learning. It's supposed to be this drug that makes you more attentive. At least more excited or excitable or caffeine.
Well, and that's exactly
only
right.
had my normal 2 cups today, I promise.
That's exactly the effect that they ended up discovering. So when you give bees caffeine, this is Sarah Arnold, who's a researcher at the Natural Resources Institute, University of Greenwich in the UK. When you give bees caffeine, they don't do anything like fly in loops, but do seem to be more motivated and more efficient. We wanted to see if providing caffeine would help their brains create a positive association between a certain flower odor and a sugar reward. It's really quite challenging environment out there for bumblebees because they don't have extraordinarily sharp vision at long range, according to Arnold. They need to rely on a lot of cues, such as their sense of smell, to find good flowers. So instead of dosing flowers with the caffeine, what they did was they went to the bee box. They went to the hive, and they created a mixture of sugar, caffeine, and this scent of I think it's some kind of a strawberry plant flower. Where is it at? Anyway, some sort of strawberry. And then they watched where the bees went. And they had some trained bees, some untrained bees. They had all these groups, and they went out and tested them. It turned out the 70.4% of the caffeinated bees visited the strawberry flowers first, which was much higher than chance. It was compared to 60% of the bees given just the strawberry odor and sugar, but without the caffeine. And it was almost 44.8% of the bees given only sugar flew to these strawberry flowers first. So the suggestion was the caffeine did give a noticeable edge to the bees' ability to recognize strawberry flower from its odor and to remember the desired nectar. Now, this died off pretty quickly because they still had the sugar was still back in the hive. So every time they went back, they still got sugar, and then they eventually we'll just go anywhere. Oh, I should just stay here. This is fine. I don't even need to go out now. Mm-hmm. But they think this could have an implication for training bee boxes for farmers. Farmers, there's not enough. I think there's some crazy number, like 80% of the bees in America come to California for the pollination season. And so you see these bee boxes out there in the fields next to the orchard or the sunflowers or the strawberries or whatever is out there growing. But if the farmers can then train their bees to prefer their crop and maybe give them a little caffeine so they'll be better workers. This is why every office should have a coffee maker in it. Every place of work should be providing free coffee. I know
and
you're
allow
thinking,
coffee breaks.
I know you're thinking, hey, I don't want to pay for coffee for all of my employees. But the thing is, they'll be more efficient, enthusiastic, and frankly, just awake. Mm-hmm. I mean, farmers are going to start giving it to bees for free. So,
well, Justin, if you give a bee coffee, mm-hmm. they're going to ask for milk. And if you give a bee milk, they're going to ask for sugar. And if you give a bee sugar, they're going to ask
for a cookie.
For a scone. Yeah. And if you give a bee a scone, they're going to ask for gel. Should I keep going?
No, that's okay.
That's fine. That's fine. Bees can have all those things. What do you got against bees having scones?
Honey. They have honey with scones. There you go. Perfect. Full circle.
I love it. We've known bees are very intelligent for a really long time. So it's interesting to see that a compound that is known to accentuate these engagement, attention, kind of learning behaviors in humans, that it has a similar effect in an insect like a bee. So now, drink your caffeine and be busy as a bee. Mm-hmm.
Get nice and buzzed.
Mike Shoemaker.
That is the topic. The caffeine, though.
Wins the
I was going to say
puns.
Does he?
Bee-rista?
Yeah.
That's the best one.
But Michael Pollan has an audiobook all about caffeine. So this fits in here 100%.
I'm sorry, Shoemaker. I thought Bee-rista was a fabulous pun. Just want to be on the record.
I mean, somebody also said Starbugs, so that's also very good.
It's just Starbuzz.
Starbuzz is good too.
All right, everybody.
I think that we could go with that one. I think that'll catch.
I have news for everyone. Big news.
Yes.
The human genome has been sequenced.
Again? Wait, what? Didn't we do this?
That's so
or is it just like, did we just do a rough draft the first time?
Yeah, didn't it finish in the, I want to say late 90s we finished it, right?
No, it was early 2000s.
Oh, okay.
Yeah. 2013 was around, 13 was around when they announced the completion, but it had a whole bunch of holes in it. And then around about 2019, some researchers decided that they were not happy with the holes in the DNA. And it wasn't a lot. It was about 8% of the genome. And computational biologist at Johns Hopkins, Michael Schatz, says there's basically an entire human gene chromosome that had gone missing. So, of course, researchers are like, what's in there? What's going on? And Adam Philippi, a computational biologist at the National Human Genome Research Institute, one of the researchers, along with Karen Maiga, a geneticist at the University of California, Santa Cruz, who founded the Telomere to Telomere Consortium to fix this problem, said, and this is from Carl Zimmer's article in the New York Times, that part of the motivation for such an audacious project was that the missing gaps annoyed him. They were just really bugging me. You take a beautiful landscape puzzle, pull out 100 pieces and look at it. That's very bothersome to a perfectionist. So because there were bothersome holes in the data for the DNA, for the human genome, these researchers pulled together not long ago, 2 years ago, to complete this. And they got around 99 scientists working to sequence what was left and actually sequence everything. They figured out new ways to make sense of the data, to figure out how overlaps were working, and to just to work it through. So it's not just that they filled in the holes by just sequencing what was missing. They did sequence the whole genome, and they put it together, and they ended up finding about 2,000 genes that we didn't know of at all. A lot of them don't look like they work. They look like they have mutations, but there are about 100 or so that might be able to produce proteins, and we don't know what they make.
Important.
Yeah, and so now researchers are looking at this new data, and they're starting to take, instead of matching up their patients' genomes to the old version of the human genome, they're now starting to match up their patients' mutations against this new data to see if there is anything new that comes of it that can help them treat diseases, that can help them understand where certain diseases that are genetically based come from.
We should talk to the guy with the really big protein that he can't get into the AlphaFold, because he's been like, it's a human-made protein. They're like, no, it isn't. It's not anywhere in the genome. He's like, but it's in the humans. They must have gotten there somewhere else. No, I'm telling you, it's really big. It's humans. And now, finally, he's like, aha, I've been telling you all for many years.
Yeah.
Nobody listened to me.
Nobody listened. Yeah, they found some weirdness around the centromeres. With the centromere, that part of the chromosome that kind of moves things around during cell division. It anchors proteins during that division process. And apparently there's a lot of repeated stuff in there, but there's a lot of DNA that they don't understand why it's there, because it doesn't seem to be involved in the centromere's key purpose. So there's a bunch of stuff that's kind of popping up that they didn't know, that now they're getting a little bit more information. And so this is very interesting. So yeah, 2021, we have the entire human genome. Finally. Yeah, it's pretty good. It's exciting. Yeah. I mean, maybe someday we'll actually know what to do with it and how to interpret it. That's the next step. This is This Week in Science. Thank you for joining us for another episode of science news and discussion. If you enjoy the show, sharing it with your children, sharing it with your family. Yeah, you can share it with your friends too. Tell them about TWIS today. All right. I only have a couple of COVID stories this week.
They're not good. They can't be good.
Nope, I just want to follow up on the CDC's reassessment and the fact that they really are saying, yes, if you're vaccinated and you're in an area of high viral spread, then it would be safer for you. They're recommending that you wear a mask in public places, indoors, where there are other people. The Delta variant is about 1,000 times stronger than the previous variant. So there are calculations that have been made that, of course, are being contested because it's not exact, but the CDC had said you could spend about 15 minutes, if you're unvaccinated, wearing a mask indoors with another person who was wearing a mask indoors. And that was because of the amount of viral particles that are released during breathing and how many particles it seemed to take to cause an infection. They said about 15 minutes and you'll get infected. Well, based on the fact that it's about the Delta is about 1,000 times stronger, that now comes down to about 1 second. You can spend very little time with somebody and still get infected because there are 1,000 times more transmissive particles being released with the Delta variant. This is why it is more transmissible. There's just more of it. It's why more people are catching it. It's why more people are transmitting it, because it gets in. It's just making lots and lots of viral babies inside your nose. We don't like that.
So wear a mask indoors again.
Again, please.
And
because
encourage your friends and family to be vaccinated. Please.
Please. Yes. And apparently there are people who have been suggesting that you don't need to get vaccinated. You can use Ivermectin, which is an anti-parasitic drug that is used very, very well, very often for getting rid of parasites. It had never been used against COVID before, but there was some people said, well, it works here, and there was a little bit of data, and so individuals, because it's a very inexpensive drug, people have been pushing for the use of ivermectin as a treatment for COVID-19. The Cochrane Collective, they review ideas. They go and they review all the literature, and they do these amazing reviews, and are like, people look to them in the field. What did Cochrane say? They did the review, and the Cochrane review has come to the conclusion that based on the data, laboratory tests suggested a weak effect on SARS-CoV-2 virus in a test tube, but did not seem feasible in humans as the doses needed would be large. However, early small trials suggested large effects on mortality, and this has led to some advocacy groups lobbying for its widespread introduction worldwide. And in the published paper, the researchers go on to say, the coordinating editor, Professor Paul Gardner, went on to say that this is a great review from an experienced team. The hype around ivermectin is driven by some studies where the effect size for ivermectin is frankly not credible, and this has driven the conclusions in other reviews. The study with a huge effect has been retracted as fake. Careful appraisal is the cornerstone of Cochrane's work, and with such extreme public demands for a drug to work during the pandemic, it remains vital that we hold on to our scientific principles to guide care. And so the main authors went on to say that current evidence does not support using ivermectin for treating or preventing of COVID-19 unless they are part of well-designed randomized trials. And there are none of those trials here in the United States. Oxford is going to be starting to use ivermectin in part of one of their trials, and I believe there are a couple of other ongoing trials as well. But for right now, don't try and rush things that you do not know, Jon Snow.
My mouth is very small.
I know, I'm watching your mouth, and if the listening audience could see what has happened.
My mouth is very small because
very
I'm very
pursed.
angry because people would rather catch COVID and use experimental medicine than take a vaccine that has been widely tested.
Yes.
It's very frustrating to me.
No,
very
but it's just
frustrating.
business. I mean, they don't have a touch. They don't get a touch the vaccine money. But they got all kinds of sideways cures that they got stakes in. That they get a nice touch. And so, yeah. Hey, I got a suggestion.
Yeah.
Before we do global pandemic ×2 and need a new vaccine, can we go back to my first suggestion, a year and a half ago, and just take 8 weeks off?
I really could use
everybody.
a vacation. I'm tired of my house.
Everybody.
Yeah.
Take 8
would
weeks
be
off.
ideal. Yeah.
Everyone stock up.
Everybody. Otherwise, guess what? It never ends.
Yeah. No, in fact, it
just
gets
never
worse
ends.
again. Yeah.
Yeah.
I don't want to do Christmas over Zoom again.
No.
Actually, that part was fine.
Christmas and Hanukkah and
I don't
everything else.
I don't really want to. I no longer want to hang out with people. This is what I've learned from the pandemic is that, you know what? Turns out when I don't interact with you all in person, it's fine. It's just fine.
I'm kind of okay with it too. I would like to go more places though. I am getting.
I will go on record as being not okay with it.
Yeah. Okay, 8 weeks.
I would like to hire
8
a
weeks.
babysitter again.
We'll call it for like 2 months from now.
Mm-hmm.
Everybody stock up. We don't need 2 months. We'll do it in a month. In next month, everybody stock up for this month. Buy the things, all the toilet paper that you need for 8 weeks. Not 8 months, 8 weeks of toilet paper. Buy all the stuff, gather it. We're gonna have a moratorium on rent. We're gonna have the unemployment things go out every. Just
Ooh,
nobody.
and then we can
leave.
just like start everything over. But this is after
Start
show
the world
ideas.
It's got to be on the show, otherwise they don't hear it though.
I know. All of the amazing ideas. Y'all know that this show only happens because of you. This is This Week in Science. Thank you for being here and being with us for this episode. We hope that you're here next episode, and we hope that you were here the last episode. We hope that you stick with us. And if you would like to help support TWIS, head over to twis.org, click on the Patreon link, and then join our Patreon community and choose your level of support. $10 and up, and you will get a thank you by name at the end of the show. And there are some fun little gifts that get sent to you as well. We really can't do this without you. Thank you for your support. All right, we're going to come back right now to a little part of the show that's called Blair's Animal Corner.
With Blair.
Why do I always turn the volume down?
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I'm gonna let you pick, Justin. You want pterosaurs or
Yes.
do you want baboons and
You had me
fitbits?
at pterosaurs.
Okay, great. You got it. So this is a study looking at baby pterosaurs. Yeah.
Who's your little dinosaur?
Yes,
Oh, you little dinosaur.
So researchers from the University of Portsmouth and the University of Bristol, along with paleontologist Darren Naish, found something about baby pterosaurs. So we haven't really found, the just science in general, I'll say, has not found a lot of baby pterosaurs, a lot of pterosaur eggs, newly hatched pterosaurs. These things have not been recovered in fossils very much. And so this is something that is more of a recent discovery. The first fossils of pterosaur embryos were discovered in 2004, but they've known about pterosaurs for over 2 centuries. So all that to say, exactly the biology of a baby pterosaur, how they come out of the egg, has been a bit of a mystery to this point. For those of you that don't know, pterosaurs lived during the Triassic, Jurassic, and Cretaceous. That's about 228 million years ago to 66 million years ago. They were around for a long time. And so there was this question of what baby pterosaurs looked like and if they were capable of flight. Because if you think about birds, for example, they spend a pretty long time like wet, gooey, and then fluffy and just completely helpless. It takes them many days to weeks, depending on the species, to be able to fly. So there's this question of, do they come out helpless? It's called altricial. Or do they come out precocial, like a baby cow or a baby giraffe that's like up and running around in an hour, right? So this question was actually answered via fossils. And so they were able to look at humerus bones of these babies, and they found that
Those
they
are
were
the funny
stronger.
bones.
Yes, the funny bones. They were stronger than that of many adults. So they were strong enough for flight. So then they used computers to model the flying abilities of hatchlings, and they used previously obtained wing measurements from established hatchlings. So these are like juvenile pterosaurs. Then they had the embryos, and then they had the adults. And all of this compared together found that as I mentioned, strength of the humerus bone was more. It was enough that they feel very confident that they could fly, but that they were flied very differently than adults. So these newborn little hatched pterosaurs were about 25 centimeters long. So that's like under a foot. It's a very small, it could fit in the palm of your hand. Spill out a little bit, but pretty much could fit in the palm of your hand. And they were strong enough, based on this computer modeling, to sustain flapping and takeoff, which is pretty stressful on a body. And they were shaped and powered for kind of flapping flight instead of gliding flight. And so adults do more gliding flight. So they think that parents, the adults, were slower, and the younger... Oh, sorry. I got that backwards. So the hatchlings were smaller, or were slower because they're smaller. So they just get less power, but they were more maneuverable. So the adults, being these big gliders, like a hang glider, it takes forever to be able to turn, right? So they're faster, but they're less maneuverable. So the question is this just the way they come out and the fact that they're smaller and their muscles, I don't know, maybe they're not developed the same, or there could be all sorts of reasons that they fly this way? Or is this actually the result of evolution? And did this enable them to rapidly escape predators because they're so tiny and they're easily preyed upon? So then that leads to other crazy questions we can never really find out, which is, were they independent from their parents? Because if they were, they would need to be more agile because they weren't being protected. Did flight style influence habitat choices? Did they separate from the egg and the parents and go into a different type of habitat or a different niche because they flew differently? Did this change as they grew? So there's all sorts of potential kind of further conclusions you could draw from this research. And so obviously you can never just observe a pterosaur and figure this out, but you have to kind of piece it together with other clues from the fossil record.
Well, the thing that makes me wonder, right, is did the adults continue to be able to fly? Like, did the adults grow to such a size where they're just like, it doesn't even make sense for them to fly, but it's such an advantage for the children to be able to be small and get up into the air away from predators that, because the parents are pretty decent size. Like, it's even hard to imagine a pterosaur flying. Like, it's just like, it's almost like that can't be a thing that ever existed big. Mm-hmm. Maybe they didn't fly. Maybe they just came back, that's just a juvenile thing, all the flying.
I think they'd get eaten real quick because they'd be really clumsy on the ground. They'd be lunched real fast.
They have an almost quadrupedal ability to maneuver on the ground. I doubt they're very quick, but they would run into that anytime they were on the ground. I don't know.
And for those of you listening, the chat room is telling me that I should have made very clear. I'm talking about pterosaurs.
You just blew up your mic too with the hard P.
Sorry. Pterosaurs. I got my shield up.
It's my fault actually, because I was like, "Oh, he's a cute little dinosaur," and they're not dinosaurs. So
It's anyway. It's with the P in the front, like pterodactyls, but there's a whole family of flying dinosaurs. You should have caught on by now. I was talking about flying dinosaurs, right? Like I feel like that
flying dinosaurs.
I thought they weren't flying dinosaurs.
They're not. They are and they aren't. It's kind of like saying a spider's not a bug. Okay.
It's like saying Curious George is a monkey. It's not a monkey.
No, that's different.
That's the same thing.
No, you're right, actually. You're actually totally right about that. It just hurts my heart so much more. Anyway, so that's the pterosaurs. Moving on. Yeah? Let's talk about baboons and fitbits. So this is a study from Max Planck Institute of Animal Behavior. And they put what is basically fitbits, accelerometers, on an entire troop of baboons, about 25 baboons. And they wanted to see the detailed movement and behavior and related energetic costs of a troop. This is because baboons live in stable societies of up to 150 individuals. They can be as big as 30 kilograms, which I'll let you guys do that math. I can't do that in my head. I can do some of the other stuff, but not this one.
So 30 kilograms is approximately the same as 30,000 grams.
Very good. Yes.
And it's also, I did a quick Google while you were doing that. Thank you very much for the vamp. That is 66 pounds. So anyway, they can be as big as 66 pounds. They can be little babies. So all that to say, they have this mixed age group. And as you imagine, this is directly related to our last story, that 66-pound baboon and a baby can move at different speeds. So they wanted to see the dynamics of the speed and pace and energetic output of the individuals in this troop. How do you move as a troop of 25 or potentially up to 150 when you have this wide range? Do you all slow down to the slowest? Is there kind of a push to get to a middle ground? Is everyone following the dominant male? What is happening there? And so by being able to strap these accelerometers to all of the baboons, they could figure out the energetic cost and the speed and the relative location of all these guys. They actually studied this baboon troop since 2012. The senior author Meg Crowfoot has been studying them for almost a decade. And so they came up with about 10 million points of data showing where the animals moved, and the accelerometer had 120 million readings, giving ultra-high resolution information on movement behavior. That's like speed and the number of steps. So while the sample size in terms of individuals was not large, the actual number of data points over almost 10 years is very large. So there is something that you can draw from this and kind of get a nice average, at least for this particular troop of baboons. And they found that the animals do in fact have a preferred speed of movement depending on their body size. So a large male with longer legs will obviously move with longer strides, that nice pendulum motion, require fewer steps to get farther. So they like to go faster. Juveniles have smaller legs. They move slower. Anyone who's tried to walk hand in hand with a small child knows this. So knowing all that and being able to come up with their preferred speed based on energy output at speed and things like that, they then looked at how fast the troop actually went. And it looks like all baboons compromised so that they could match the pace of their nearest neighbors. This sounds to me almost like a school of fish, which seems very cool, kind of population dynamics, and there's all sorts of interesting stuff happening there. But when the group spreads apart, they try even closer. So when they span farther distance as a troop of 25, they kind of jog to catch up with those around them even more. And so this provides evidence of what they consider a democratic process at work, but really I think it's more just kind of mechanics of a group in movement. And so they specifically wanted to see too what the dominant male was up to, because in one-on-one interactions, dominant males definitely have the ultimate say-so, but in collective movement, they actively slow down to stay with the rest of the group. And it looks like the individuals that sacrifice the most in the group are the smallest ones. So the smallest baboons.
Because they have to keep up, right?
Yes.
They have to work so hard.
Yeah. So they have to keep up to get with the rest of the group. They have to travel longer than they wanted to go, and they put out more energy. They pay a disproportionate cost to stay with the group cohesion. So the researchers say this might be because they have the most to gain from group membership, which totally makes sense. But I also think the youngins can handle more physical stress, and that might also have something to do with it.
And it builds character, even if you're a baboon.
I will say though, yes, you have the young'uns with all the energy. They can keep up. That's great. But they also tire out, and then they whine, and then they end up dragging on you, and you have to carry them. And
but then they also recover the next day like it never happened. Whereas you are still tired from the long walk in the playground and the thing.
But as a vertically challenged person, slightly, I'm not as tall as some of the other people I know going walking with. I have always had to, my legs, they're shorter. I've always had to move them 2 strides for other people's 1.
Now, did you keep up with those people, Kiki, because you are worried about being predated upon?
It depends. I was going to say no, but then I thought about places where I'd been, and I'm like, well, actually, that depends.
Yeah, walking home late at night is a whole different story. But anyway.
Some neighborhoods, yeah.
So the last bit of this study, which I think is interesting, is they used computer simulations to see how the group would look if everyone moved at their preferred pace. What would happen to this kind of dynamic, this flow of the group? And then they compared with individuals adjusting their speed in response to others. And they really showed, to no one's surprise, I'm sure, but the science has to be done, right? That the locomotor compromise is a requirement for cohesion, which just means somebody's got to speed up and somebody's got to slow down if everybody's going to stay together.
Yep. You have to come to some kind of agreed upon middle, and it has to be, At some point, I'm sure if there is a predator and one of the large males or the sentries sees that male and is like, or sees the predator and is like, everyone get together and go faster, then everybody runs faster together. But it's all about protecting as many of the group as possible.
Yeah. And so last thing I'll mention is about kind of next steps of this study, the implications, which I think is an important thing to mention here. So the differing locomotor ability, the different speed at which animals can move, might put an upper limit on how large a group can be, might force specific individuals to group together, and might have impacts on the structure of animal societies. So to this point, there's not been a lot of fitbits on animals in the wild. This is one of the first studies where they've really done this thoroughly. And so being able to study this aspect of cohesion of a group or a troop or a pod or a pride or whatever you want to call it, could have important implications in the future when you're studying the kind of population dynamics and ecology and all these sorts of things. So
and self-driving cars.
Yes, and that. Somebody's got to slow down. Somebody's got to speed up. Everyone's got to stay together. You're going to create traffic.
Yeah. You're going to get traffic without a jam. This is This Week in Science. Thank you for all those animal stories, Blair. It is time to turn now to Justin Science Corner. Justin, what you got?
I've got an international team of scientists.
In your back pocket.
Yeah, locked in the basement.
No.
International team of scientists has found that changes to human oral biome during the shift from foraging to farming were not nearly as dramatic is that those who have occurred in modern times due to antibiotics. They looked at 10,000 years of history, basically, from around the Neolithic up to the end of the Stone Age, or at the end of the Stone Age up to medieval times, and they etched out mineralized plaque, dental calculus, and did DNA checks on it, and they could tell basically what the biome of the mouth was. Interestingly, they were actually even able to track certain species of bacteria's emergence or sort of taking over or being eliminated from the mouth over time. But really the changes were pretty minor until we get to the modern age and we have antibiotic use, and maybe the toothbrush starts getting used more frequently, toothpaste, that sort of thing. And now there's some antibiotic resistance in microbes, and there's much more significant evolutionary changes taking place within the bacteria of our mouths in the modern age. So this is going to be 22nd century problems. So I don't think we have to worry about runaway antibiotic resistant mouth microbes yet, but it may be coming. And then here's the one I'm going to end my segments this week with some terrible, awful news that has no silver lining and should give listeners a knot in their stomach as they realize that a living nightmare from which they cannot wake up is going to be the new normal.
And it has nothing to do with antibiotic resistance in your mouth.
Nope. That's going to be 22nd century problems.
Okay.
If we get there, for those who get there.
Okay.
This is the global approach to climate change has so far been, say you're going to fight it, make some bold policy change suggestions, stick to a few of the easier to reach goals, congratulate yourself for trying. Meanwhile, according to IPCC researchers, Earth's vital signs have continued to deteriorate. Researchers, part of a group of more than 14,000 scientists who have also signed on to an initiative declaring a worldwide climate emergency, said that governments have consistently failed to address the root cause of climate change, which is the overexploitation of Earth. Since a similar assessment in 2019, they noted an unprecedented surge in climate-related disasters, floods in South America and Southeast Asia, record-shattering heat waves and wildfires in Australia, the United States, and devastating cyclones in Africa and South Asia. They apparently put this together before the unprecedented flooding in Europe, but that will be next year's report, no doubt, alongside something even worse that we didn't even realize was going to happen, which is also going to be new normal. Of 31 vital signs that they were tracking, key metrics of planetary health that include things like greenhouse gas emissions, glacier thicknesses, sea ice extent, deforestation levels, that sort of thing. They found that 18 of those 31 hit record extremes for the first time in this past year. For example, despite the dip in pollution, thanks to the pandemic of 2020 and 2021, levels of atmospheric CO2 and methane are at all-time highs in 2021. Greenland and Antarctica both recently showed all-time low levels of ice mass. Glaciers are melting 31% faster than they did just 15 years ago. Ocean heat and global sea levels set new records since 2019, and the annual loss rate of the Brazilian Amazon reached a 12-year high in 2020. Things are so bad in parts of the Amazon that the Intergovernmental Panel on Climate Change found forest degradation linked to fire, drought, and logging are causing parts of the Brazilian Amazon to now act as sources of carbon rather than removers of the gas from the atmosphere. Livestock, such as cows and sheep, are now at record levels, with a mass exceeding that of all humans and all wild land animals combined. We are now a planet of domestic food animals. If you came from another planet and looked at what's the predominant earthling, it's domestic livestock. recent record-breaking heat waves in the western United States and Canada, researchers say there's mounting evidence that we are nearing or have already crossed a number of climate tipping points from which the idea is there is no return. These include melting the Greenland and West Antarctic ice sheets, which may now be irreversible on a centuries-long timescale, regardless of how or if humankind slashes its emissions. There is increasing ocean deoxygenation and warming waters threatening warm water coral reefs, upon which 500 million people rely on for food, income, and storm protection, let alone the oxygen produced to the planet you can breathe. Given these alarming developments, this is, say, the authors of the study published in the journal Bioscience. Given these alarming developments, we need short, frequent, and easily accessible updates on the climate emergency. The authors echoed previous calls for a transformative change within 6 years, eliminating fossil fuels, slashing pollutants, restoring ecosystems, switching to plant-based diets, moving away from indefinite growth models, and stabilizing the human population. If that last sentence, stabilizing human population, does not give your spine chills, you might not have a spine. That's saying we're breaking the planet to where it can't sustain any more people. Stop having children. Maybe we could do a Soylent Green thing instead of all these cows. They've also called for climate change education to be included in school core curriculums globally in order to raise awareness. Again, awareness is one of those things that even those that in the United States, we're 72% according to that Yale poll, believe that global warming is happening. They also only hear about it maybe once a week from the media, even though this is an impending crisis that is causing all sorts of problems. So anyway,
I think that's really important what you just said, though, because, we've been saying over and over every week on the show, tell your friends and family to get vaccinated, tell your friends and family to get vaccinated. That has replaced the usual echo that we talk about, which is talk to your friends and family about climate change. Because there's an expectation, right, that you're hearing about it enough. We don't need to talk about that right now. But that's exactly the kind of statistic that proves that you need to talk to your friends and family about climate change. Because if they're not hearing about it from the media, they need to hear about it from you. And believe it or not, there is a push-pull there. And if everybody starts talking about it and is looking for that information, the media will talk about it. Why is everyone reporting 10 different ways on the same COVID story every day? Because people want to hear about it. If we can make it where people
it's impacting their lives right now.
Exactly, yes. Yeah. If we can make it feel urgent and real and solvable, which again, COVID is, by the way, even though it's whatever. But anyway.
And so is climate change.
Yes.
Climate change is solvable as well. And I think that is
people want to learn about it.
People want to learn about that, but we also need to inform people. People need to be informed that change, it can come from them, but they also have to demand it of corporations, manufacturers, politicians. The entire system needs to change. So it's maybe it is a big ask to do it in 6 years, but there's a lot of political will that has to be addressed there. But I don't.
So again, the political will is it's of
but if we can get manufacturers, investors.
Money's make believe anyway. You want to get something done. It is.
It's make believe, but it gets people to do things.
Yes, I know.
It's why everything is done at all right now. Like, politics is driven by money, greed, and money. So if we just get people, give them the money to do the good things. Why can't we do that?
Fake meat alternatives, all of a sudden is like this multi-billion-dollar business. And it's because someone realized, hey, I can actually make this taste not like rubber. And
you know how they did that?
How
they did that? Yeah. They did that with science.
Yes, with science.
But there's so now there's this big push. And that's exactly what we need is we need people demanding things so that then the people who have the unjust, undue power will make the right choices.
Yeah. Yep. And like you said, it's a well-known problem for humanity that we have a hard time connecting with things that are not affecting us right here and right now. It's like, I'm going to put it off. I don't have to think about that
So,
right
but-
now.
and this
I'm
is
not
one
going
of
to
the
worry
things,
about
too, that-
packing until the night before I leave.
Night
Yeah.
before, morning of.
Morning of.
But,
Exactly.
But the state, the U.S. territory, at least, state, that is in the most danger from global warming and is feeling its effects probably the strongest is the state that keeps electing people who don't believe in global warming or deny it publicly, the global warming science behind climate change. So that's Florida. So there's this other reverse tipping point, I think, for at least for this country, which is when the water's high enough that Florida is underwater and no longer can sustain a population, we then can move forward.
We'll see. We'll see how that works.
Too. It's sort of
But that's not going to happen in 6 years. So let's move forward.
If you live near a coast, you're much more likely to believe the climate science, which is an interesting thing. Which is also that's where most of the population is. That's why the numbers are high. Anyway.
Yep. This is This Week in Science. That was, I mean, reality sometimes is hard, right? But we have to face it, and I'm glad we're facing it together. Let's face the science. Let's face understanding what the world is really like and move forward. And you know, sometimes the world brings us very interesting science that can distract us a little bit for just a moment from some of those depressing real world thoughts. Like, do you ever think about what happens when you move your head from side to side really fast and you're looking for things? There's this idea in neuroscience that, like, if you're moving your head really fast or there's something in motion as you're moving your head, that basically your eyes are taking little snapshots and then piecing them together, that there are these little breaks in the actual pictures that the eye is taking. That's not true. It's not true.
I've
It's
never
not
heard
true
of
at
that.
all.
So good.
And it's actually, there is your eyes like smearing the light. And that is according to a new study out this week in Science Advances magazine. According to their abstract, or the abstract of the article about it, the in focus about, which is great. This is a vision story in a section of the magazine called Focus, which I find very punny. Visual information is continuously sampled from our environment, even as the eyes move, which helps the visual system create a stable view of the world. So when you're moving your eye, it actually, like this, it's smearing things. It's taking a constant picture, and those streaks across the visual field actually help the brain to create a continuous and stable view of the world that doesn't break up and break apart. And so we're able to be perceptive, be aware of the motion of objects in our field, in addition to the movement of our eyes. But I find it very interesting that we used to think that there were these, well, our eyes do these saccade motions. When you move your eyes really fast, especially like if you're sitting in a chair and you get spun around in the chair, your eyes don't just track with your head. They go forward and back and forward and back and forward and back and forward and back. Yeah, bop, 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 bop. These saccades, yeah. And so because of those saccades, they didn't think that we got that consistent, continuous motion. But this new study by Schweitzer and Rolfs demonstrates that the motion streaks help the oculomotor system correct for variability in eye movements and help to build that stable view of the world. So even though the saccades are there, they're not really there in the way that our brain interprets it, because the light never changes. Our eye is continuously taking that picture. So they used a novel high-speed projector, and they manipulated a visual scene during eye movements. They had the eyes moving and the visual scene also changed. And participants saw 6 small patches with unique patterns. One of the patches was designated as a target for the eye movement. You're supposed to try and focus on the target. And the pattern ensured the patch would generate a unique motion streak. And then they wanted to know whether the participants were unconsciously processing the motion streaks. So they rapidly shifted the position of the target while the participants' eyes were in motion. And as a result, the eyes landed on a location between the actual target and one of the other patches, because the eyes were like, wait a minute, that moved. So the eyes didn't quite go where they were supposed to. And so once the movement was complete, all the patches were masked so that there was really no difference between any of them. And so there were corrective eye movements that had to be made by the eyes. But all of these things together, the end story for this is what I told you at the beginning. Our eyes are taking a continuous picture of the world.
I love it.
And they're streaky and smeary.
I guess you would assume based on how the eye moves that it's like pasting a bunch of overlapping photos together.
Yes.
But it's really like taking a pano.
Yeah. Yes.
So something interesting, like if you watch ballerinas doing like their spins, they're picking a spot and then they're
focus
on
the
same spot as every time they come back to sort of stabilize.
That's so they don't barf.
Right. No, I get it too. It totally makes sense. But then like everyone's like the Winter Olympics will happen and then the ice skaters are doing that like blurry spin where they like sort of just disappear into this hub of a spinning top. How in the world do they do that? Because that right there, that seems like that would be just so utterly disorienting.
Well, it's training, number one, but it's also...
So they don't just know how... This is what...
the way that it's set up, though, because the eyes are not kind of going back and forth and taking these individual shots, if you would imagine, if they were taking the individual saccade-type shots, the ballerina would have a much harder time being able to find that target at the other, when she came back around 360 degrees, because the pictures being pasted together by the brain...
It wouldn't make sense.
It wouldn't make sense. It would be hard for the brain to make out, like, where should things be, and am I in the right place, and is this the right thing? But it's specifically because of the streakiness, that streak goes around, and so you still have a streak that continues from that target, from where you started to where you're ending. So your brain is like, there's this line of continuance, and I've been following it. So that's how it helps with the stability.
Okay, that might be true. Now what I want to do is I want to blindfold some ballerinas.
Yes.
And set them spinning on a stage and see how much of it is vision and how much of it is just like physical body memory. If I do this, I do that, and then I should be looking in the right direction again. Like how much is it visual cue? How much of it is just within?
This is the body. It's all oculomotor. It all goes together.
Just put lots of pillows on the floor so nobody gets hurt.
Lots of pillows. And my very last study for the night is about little baby dreamy mice. Little dreamy baby mice.
Oh, what is your little baby mouse?
Yeah, so this is maybe different with the way that human babies work and our eyes work, because our eyes don't work for several months after we're born. Like our eyes really they're not ready to be there. But a lot of newborn mammals open their eyes right after they're born and has to start making visual sense of the world because predators are around. A lot of precocious animals really have to do it very quickly. But these researchers publishing in Science out of Yale University this week suggest that there's a kind of activity in the retina of the eyes of little baby mammals. And they did this study using mice that is like dreaming about the world that they will live in. Yes. So before they're born, these little mice have waves of activity from the neonatal retina. This is before the eyes have ever opened, before they've come out of the womb, before they're still in there, waiting to be born, and they have these waves of activation. And these waves of activation mimic actual light-stimulated activity that would occur if the animal were moving forward through its environment.
Wow.
But how could this be?
How could this be? This dreamlike eye activity.
Is this something that's being provided somehow? Is this something that just was happenstance and gave an evolutionary advantage? This seems insane.
Right. I mean,
it's practice.
It's practice. It's like the
It
ballerinas, but it's before practice. It's practice. But this is the interesting aspect. And I want to know, this is in mice. I'd love to know if this is something that happens in neonatal humans as well. Mm-hmm. But, and I'd love to know if there's a different stage of development at which this is present in different species depending upon their precociousness. So which species of mammals have this activation, this preparing for use? And when does it occur? How early does it occur?
But again, how? Like, what is the mechanism? This is killing me. I'm so confused.
I don't know, but this is a huge question, right? The why is so that the animal's more prepared to view things in the world and to survive. Mm-hmm. But
Warming
the how,
getting it ready for how it's going to be.
How does it know that the eye is going to need to have these kinds of signals in it to move through the environment? This must be evolutionary.
It's
evolutionary evolved, but yeah.
Is it a genetic memory? Is because we know the mom can kind of provide weird memory-based biology to the baby. I don't know.
So remember, it seems like we always seem to have this sort of boxed, disconnected idea that the unborn child is removed from all life. It's like an alien showing up on planet Earth for the first time.
Like
I don't even know how to breathe. What am I doing here? But they are part of a billion-year-old life form that has been surviving on this planet. Now we bud off into these separate individuals that are so different from each other and think we're unique. But ultimately, we're all the same organism that's just sort of mutated off into different directions and had a billion years of maneuvering on the planet. So somewhere within all of that information of existing on the planet, the eyes know what it's going to be facing.
And the brain as well. And so this is part of it, which is the brain before animals are born already has started firing and wiring and starting to put things together. And so this is probably part of that process of the amacrine cells within the retina firing and trying to wire with the brain. Mm-hmm. It's probably setting up those neural circuits for success. And what makes me say that is that the Yale team also removed or stopped. They blocked the function of these amacrine cells in the retina. And so the flow of activation did not happen in those baby mice. And it impaired the development of the ability of these mice to respond to visual motion after they were born.
So you said endocrine, yes?
Amacrine.
Oh, amacrine. But that's still a hormone-related system.
Crine, yeah. So it's related, but it's
so this is my new hypothesis based on very little.
Hold on.
Yeah,
Go ahead.
And then see what an amacrine is in a human, because they name the hormones different things even when they do the same stuff.
It's not hormone. It may, because of the compounds that it releases, maybe it has the INE ending, but these are interneurons.
Okay.
In the retina, so they connect.
Okay. My question is,
cells.
Are the embryos dreaming when the mom is dreaming?
Oh, I don't know.
Because this is now my follow-up, right? Is there some sort of signal getting pushed to that being to promote that?
Yeah.
Or is it separate? Yeah. curious. Just,
But to some great degree, too, that is the same being.
right. Which is why it might be related.
Yeah, well, they are related.
They are, but is it? Anyway, I don't know. It's the mystery of life. The miracle of life.
Mystery miracle.
Mystery of life.
Still a
at
mystery.
last I found
Mysteries.
you. Mystery of mysteries. Yeah, but it's
This
really.
is Mario Alonso you're belting out there.
But the take-home message from this is that the brain, the organism, the baby is dreaming about being born, is preparing to be born.
I don't want to go out there.
No, it's getting ready. It's like, yes, let's go. I'm going to know how to see. I'm going to do all this stuff. I'm practicing. I'm going to see the bright lights. Bright lights, big city. Here we go.
No, I was way more comfortable in the womb. Yeah, it was way better.
Yeah.
Yeah.
Try again tomorrow.
Have we gotten to the end of another episode?
I think we have.
Woohoo! We did it. We've gotten through the science. We didn't talk about the very old sponges. Maybe you out there will have to go.
850 billion-year-old sponges.
Billion-year-old sponges.
Yeah, like the oldest sponge life ever discovered, and it's in Canada.
Oh, earliest animal yet found.
At the top of a mountain.
890 million years old.
You know, rocks. Yes. Not billion. Million, yes. Old life.
Oldest animal on Earth by more than 300 million years.
And really, they're just looking at these little structures, these little kind of what look like could have been filaments in rocks. It's like
Little spicules.
yes, it's amazing.
I didn't see the pictures of the sponge one. I pictured it like a sponge imprint on a stone.
Yeah, it's like a sponge imprint, but it's
Yeah,
yeah.
but it's the grippy feet.
Was it the yellow side or the scratchy green side? That's really the question.
Neither, Blair.
Oh.
All right, now we're really done.
Okay.
We've really done it now. Thank you for being here for the show. We hope you enjoyed it. Thank you to Fada for your help with show notes and social media, for helping with those YouTube trolls tonight. Gord, Aaron Lore, Hot Rod, so many others. I know I'm going to miss someone. Our moderators over on Twitch and on YouTube, other places. Thank you for modding. Identity 4, thank you for recording the show. Rachel, thank you for your assistance and the editing. And I do want to say thank you to our Patreon sponsors. I need to change screens. Wow. Un momento. I had it all set up, but then it didn't follow me, so I'm sharing it now. Thank you to Pierre Valazar, but Ralph E. Figueroa, John Ratnaswamy, Carl Kornfeld. Whoa. Kira, Carl Kornfeld, Jen Myronik, Melanie Stegman, DeKramsta, Karen Tazi, Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vigard Shefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Parangor of Sharma Shoe Brew, Don Mundus, Stephen Albrand, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Ked Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflog, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood. This profile name is hilarious in the context of some other podcast. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, please head over to twis.org and click the Patreon link on next week's show.
We will be back broadcasting from our YouTube and Facebook channels and from twis.org/live.
Hey, do you want to listen to us as a podcast? Whoa, I'm going to do that again.
You can't. It's a live show. There's no doing it again. You're still doing it.
Hey, do you want to listen to us as a podcast?
Yes.
Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, you can get your friends to subscribe as well.
Yes, and for more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can sign up for a newsletter.
You surely can. You can
Or
also
ballet
contact
classes.
us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, and Blair at blairbaz@twis.org. That's lots of ats. Just put TWIS, TWIS, in the subject line, or your email will be spam filtered into the spongocele of an 890-year-old sponge.
If there wasn't enough ats for you in the previous segment there, you can also reach out to us where we are at TWIS Science, at Dr. Kiki, at Jackson Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from this show, remember,
It's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
Boom. Pachoo. It's the after show, everyone. Hey. Blair. Are you punchy?
Yes.
Yes. Yes. Yes. In the mood.
Yes.
I think I am too.
I don't know what it was. The heat today. Being tired. I don't know. Doing statistics right before the show. I don't know. I don't know.
I know. Statistics always makes me punchy.
Yeah.
Just shake my head after all the statistics. Yes, Aaron Lore. Blair's hyperness is the Zoomies.
The human Zoomies.
The human Zoomies. Our cats do that. Like every night. There's some point at which the Zoomies happen. They run around the house. They run from one end of the house to the other end of the house. They run down the stairs. Boom, 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 boom.
They're ridding your house of spirits.
Is that what it is?
Yeah.
I hope so. Yes. I would like to go to the snack bar. Actually, I had dinner. I'm fine.
You're fine. It's all fine.
Yes. 7 cats. Oh, that's fantastic, Aaron Lore. We made it through a show even with the Zoomies. Zoom, zoom, Zoomies. TWIS time, Zoomies.
It makes the show go faster for me.
It does. Okay, I'm in. I'm here. Let's go. Let's talk. It was funny to have Justin not quite on the Zoomie level, which is probably a good thing.
What do you mean not on the Zoomy level? What are you talking about?
We decided, the chat room decided that that Blair had the Zoomies tonight. Look, there she goes.
Had?
Still has. She got the Zoomies.
Oh yeah, it's usually me who's being all like.
That's what I was saying. I was like, it was really funny. Justin was like the one of us who wasn't in the Zoomies tonight. And I said that was probably a good thing. It's always good to have at least one person.
One stable voice of reason to guide us across the waters of the science news.
Yeah, like Carl Sagan.
He had a very soothing voice. You know who I still like? So Carl Sagan is a great, like, if you're looking for a sleep aid.
He cultivated that voice.
If you're looking for a sleep aid, forget like the waves and the stuff. Like, put on Carl Sagan talking about space and billions and billions. But you know who else I really like?
Lovely.
And it's really unfortunate because I absolutely love his content as well. Leonard Susskind. Go find a Leonard Susskind. He has just this cadence in his voice. Like, I love listening to it, and then you fall asleep like you're thinking about Einstein's equation stuff, and then you're asleep. He's amazing. I couldn't take any of his classes. I think he's got too soothing, reassuring of a voice.
You'd be like, oh, it's wonderful. These black holes, they are great. Good night, Fada. Thank you very much for the hitting the YouTube and helping when it was needed. That is, your quick response was much appreciated. I didn't see what was going on until I was like, what is Blair talking about and what's happening? And then I saw something hit the chat room and I was like, that's awful.
Pretty unacceptable. I kind of feel like
it was really unacceptable.
I understand why they allow swears in YouTube chat, but I feel like hate speech should not be allowed. I feel like that should automatically be flagged and pulled.
Yeah, and
I don't know. I feel slightly reassured by it. It's always a good sign when, out of the ether of the people who could be targeted by, like, Nazi hate speech, that they chose you. It makes you feel like I'm really doing things. I'm on the right side of the universe, which would be the left side of the universe. It's kind of clear, but it's always a little bit reassuring to know that the people who are the wrongest people in society chose to target you. Because it just confirms that you're doing everything fine. There's nothing wrong with you.
That's nice that you can say that, and that it doesn't cause trauma for you to see those things.
Yeah,
but
for some people it does.
That is the problem, right? Is that it can be offensive and not be triggering or traumatizing, but to other people, those words are extremely traumatizing.
To me, it just looks like
it's obnoxious. But for some people, it's much worse.
Yeah.
But to me, it always speaks loudest of the person who's using the language.
For sure, but it's disruptive.
Absolutely.
It was incredibly disruptive.
And I'm glad we have buttons and levers and mechanisms to remove these sorts of things. That's fantastic.
Oh yes.
But I don't know. It's always showing that the level of stupid that's opposing you in this world is that utterly bankrupt mentally. Then it really shouldn't be a challenge out there.
I don't know. From my corner, I kind of feel like one thing I think Germany did right was outlawing the swastika. I think it was a good move. Mm-hmm. Not allowed. Sorry.
Say this again?
Yeah, post-World War II, you're not allowed to wear, display, or show a swastika in Germany.
In Germany. And I think that, did we actually technically ever stop being at war with Nazis? Or was it like a separate German, like the country Germany surrendered, but the Nazi part? Like, I don't know. I was getting a little bit of heat for saying this, but the whole punch all Nazis kind of a going forward. Like, don't like. Mm-hmm. There's nothing wrong with it. If anyone is trying to grab onto that ideology or symbology as a means of anything, they're an enemy of the state. Actually, they're an enemy of the world.
And all I'm saying is
they're an enemy of people.
That it would be easier for me as a human to exist and not feel attacked if that was just not allowed. And that's kind of my point, is that yes, hate speech ultimately points a finger at the person doing it as being in the wrong, but it still hurts people. And I think that's my point, is that like, why are those words allowed in the YouTube comments section? I don't know. Just, hey, YouTube, maybe make hate speech not allowed in YouTube. You have AI. You can figure out. You're connected to Google. You can pull those words and not allow people to post them. That is perfectly reasonable. Yeah. The words that were getting posted tonight were like in the top 10 list.
Oh,
of
totally.
Words that are hate speech. So that should be pulled.
Should have been easy. Yeah, I agree.
That's my point. Is like, just no hate speech allowed.
And I'm very disappointed in Gwen Stefani. Can I just say?
Gwen.
Fun fact. Yeah. Fun fact. Her and my pops were Whole Foods buddies. They would used to go shopping at Whole Foods together. I'm amazed how she's turned out. Which, okay, for full reference, somebody was spoofing Gwen Stefani as part of, and then the free software guy. I can never remember his name.
Free software guy.
The guy who wants all software to be open source. He's like a little bit of a theoretically activist for open sourceness. Anyway, they used his name and image as one of the things. So they were spoofing sites as part of this as well.
Oh,
Don't be mad at any of the names that you saw associated with those, because they had nothing to do with the real people.
Well, luckily they've been deleted. But also, the Twitch chat room is telling us that Twitch already does that. So YouTube just has to catch up, it seems like. So, yeah. It's for this year, for my birthday.
It's just right, like the guy who stormed the Capitol and then was like, she's like, I'm at the White House, which was
Yeah.
like, yeah.
Always good.
Yeah, Aaron Lore was saying that he was, he saw speaking of stuff, different things, he saw the podcast Factually with Adam Conover. The latest episode has major errors in it, and he's wondering about prior episodes of it being accurate.
Wait, what? What are you talking about?
Prior episodes being accurate. It's a show called Factually. And there are errors in it. I mean, that's a good question. Sometimes, after doing a show, sometimes errors are made. If they don't, if they don't correct those errors, that's a problem. But yeah,
we're a live show, and
yes.
and
We
doing
make
a live
errors.
show,
We try and correct errors though when we make mistakes and say,
We
"Hey,
try.
I got that wrong."
It's not really a dinosaur. It's a flying lizard. It's a pterosaurs or whatever. Like, I'm making more errors as I go. That's fine.
Stephen
We can
Rain's
have
been
those.
giving Gwen Stefani a talking to. That's funny. Well, yeah, I mean, I think there's a lot of media out there that you trust it, and I hope the same for TWIS. You enjoy listening to it. It's a piece of information, and you can go, "Hey, that's awesome," and you can confirm it if it's something.
And there's links. There's links to
and we
confirmation.
share links, and yes,
at the website.
Yes. So if you go like, what was that story? I need to make sure I can confirm that and get the details right. You can go look at the links.
There's even in the disclaimer tonight, when I was quoting from the Yale study on
Trust but
people's
confirm.
beliefs, there's even a link next to the disclaimer that you could go to the website and find, and maybe find out, read some more interesting stuff from that poll.
Oh, I'm supposed to put a link in the disclaimer this week?
Actually, what's interesting is that one of the things I didn't even get to there is that despite all of those numbers of who believes what is what, 86% said they did support funding of alternative energy. So even if you don't think global warming is real, or that global scientists know what they're talking about, or any of the rest of it, even some percentage of those people are even like, yeah, electric cars are cool. Like, of course we need that. We need windmills and solar power. Of course we do.
We do.
Like 86%, which it's higher than the number that are even concerned about global warming, are still like, yeah, solar panels. I'm down. That's fantastic. That tells you also a place to focus on this.
Solar panels, windmills.
Well, it's because, who doesn't want to harness the power of the sun? You can't run out of it.
We
It's
already
free.
do. It's how ATP works or something. Like it's like we all. That's like the basis of life.
Photosynthesis,
Yes, that's how photosynthesis works.
vitamin D.
Imagine cooling your home with the same sun that's making it hot.
And even if you're one of these people who doesn't eat their vegetables,
I love veggies.
the food that you eat, that's what they're eating. So you still need the sun to do everything.
Isn't it a wonderful thing? Like we talk about plants, we think about plants as being the users of the sun, but we harness the power of the sun ourselves through our skin, even though we try to block it with sunblocks and melanin and stuff. It's still, the sun is important to us. It makes vitamin D. It helps to strengthen our bones. It helps build our immune systems. We are powered by the sun.
Yes.
That's amazing.
So why
Not
would
to the same degree as plants, but hey.
power? Yeah, and plants. Another really good point.
Thanks. We need critical thinking. Should get a grant, but then I'd have to write the grant.
All right. During the 8 weeks where we are in the new lockdown.
Right, I'll do the grant writing while we're in the lockdown. Wait a minute. We're in lockdown. Hold on a second.
I'm in lockdown and I have to write a grant. This is worse.
Oh my goodness. Oh, I'm yawning. I'm sleepy, I think. Tired. Maybe. Getting there. Plant-powered. Cat-powered. Purr-powered. Meow. Do you know what happens this weekend?
What?
I can never not tell people.
It's your birthday.
Yep. It's my birthday this
What are you
weekend.
doing for your birthday?
I hope to do a river float. I would like to. It's going to be nice and hot up here. And
Yes. I love
Yeah.
a river float.
yeah. I'm going to get out on hopefully get out on a river. Have a nice day of water and inner tubes and kayaks and sun and soggy sandwiches.
Yes. Good.
There's always a sandwich that ends up at the bottom of the boat.
Well, what
Don't
you
eat
do
it.
is you use I guess Kai's not using them anymore, but the food pouches are perfect. Yeah, it's perfect.
I did eat those a bit when I was giving them to Kai.
They're
They were good.
good.
They're great. They're good
Yeah.
little pick-me-ups.
I get it.
Okay. So is this too
Bad for
soon
the
after
planet, though, people.
I was just about to say, you're talking about individual minuscule snacks packaged completely in plastics.
Yes, I
Okay.
stopped. I did them a
Okay.
little bit, but usually it was the
All right.
jars, glass
You
jars.
can TerraCycle them
Look,
too. You can
you-
send them back in, and they'll reuse them.
You can. However, the one worry about the planet timeout is babies. It's
Yeah, that's
just-
also true. Yeah.
Yeah, it's just
You get a full
that's the
pass.
one where
No. There's two babies, hospitals.
And hospitals.
and research to an extent in general.
Because they use
Jeez.
so
What are we gonna
much
do?
plastic in the
Everybody gets a
hospital.
timeout.
The hospital is like, you can't be reusing stuff.
No.
Ooh, Arnlor, I want that for my birthday. That's what I'm going to drink on my birthday.
Oh boy.
Ginger ale and rainbow
So
sherbet. That
let me
sounds
ask this question,
fantastic.
Kiki.
Wow. Yeah,
You don't have
now
to
I
answer
try
it.
that.
Great.
I'm just very curious. Is this a child-friendly float, or is this an adults-only float?
It will be child friendly. Mm-hmm. Child friendly float.
I've done the
Two
other. I've done the
very
one where-
different floats.
Yeah, I've done that
Very
one
different
in
floats.
inner tubes with the inner tubes where we had a couple kegs of beer that were strapped to one of the inner tubes. And
Is
then
this
we
the
were-
American River? 'Cause
Yes,
that's where,
yeah.
yes, I've done the American River a few times. Yeah.
And we were roped to it, so you would pull your rope up close to the kegs, and then you would pull your inner tube along with you, and then you could fill your cup and then push off and be. It was a bad idea.
Terrible idea.
It was not something I would advise people to do.
It's a thing you do when you're young in the
Yeah.
Central Valley, and
you go, I'm gonna float in the direct sunlight, not drink any water, barely eat any food, and
and
just
it's gonna be all
drink
day.
heavily for like 4 hours.
Yeah.
Again,
Yeah.
in direct sunlight, not
In the
remember
water.
to reapply your
A
sunscreen.
bunch of drunk people
Yes.
on the river floating in tubes,
And then
there's
drive
some
home.
rapids, and then there's occasional rapids too. It's not even like it's just calm water the whole time.
Oh yeah, the American River. We used to call it the easy float because it was like there was one little tiny bloop bloop.
No,
Yes,
this was
yes,
this was
yeah.
then this was the Sacramento River.
Okay.
We hit real rapids.
Okay, there were
hit
no
real
rapids
rapids.
ever. We called it the easy float because that's all you had to do was float and just hope that you paddled enough to get to the last bus
Yes.
at the end of the float.
Absolutely, because you pull over because you want to milk it, right? Of course, yeah, for sure. I'm going to the Yuba in September.
the
Very
Yuba's
excited about that.
wonderful.
Yeah, I did that a couple years ago. It was so
It's-
fun.
Well, I've never been to
there
the
any
Yuba
water?
River in a part that's. That
I mean, I
big,
hope
though.
so.
No, this is no. When you said this, though, like I've been to I used to go swimming in the Yuba River all the time, but it was like kind of like a big creek. Like, I don't remember. I must not have been where
go.
it was. Yeah, I was not at a big river part. Like, if you've been on the American River, then it's not that.
Mm-hmm.
It's currently 87 feet, the Yuba River. Google says.
Grouchy Gamer, you're fired.
What happened?
Oh. Well, he said TWIS is 60, not you.
Oh, how would that be possible? The implications are there. Okay. Come on. I haven't been doing it that long.
Yeah, well, I don't know.
Oh my goodness.
You know, time folds in on itself. It's fine.
Yeah. Yeah, time does fold in on itself. Mm-hmm. You're like, oh, is this my new lifetime? Oh, this is my new thing. Okay. Yes. Oh, no. I know. Sorry.
This is a typo. No, it wasn't. What did you mean to hit?
Yeah, Arnlord's dog years.
Oh, man.
Well, this show is 20 years?
Really, like 10. That's it.
Okay, yeah. So
it's dog years.
We'll cut our legacy in half and say it's only been 10 years since we've been on there.
I mean, my stint is about to be 10 years. In January.
No.
Yes.
Wow.
Yes.
It's amazing.
Yeah.
We'll have to get you a little plaque that says you're tenured.
Yes.
We should have, maybe we should have celebratory plaques.
Oh my God, I love it.
No, please no.
Yes.
Oh,
Like,
that's right.
Can we also do Employee of the Month?
Oh, yeah, okay. Yeah, one that you can win. How about longest sustained rant without anybody else speaking?
Oh,
That could be the plaque. But I forgot you're into that. You've got like the wall of trophies.
Oh yeah, I got a new one. I got a new one before I retired from bowling.
You did not retire from bowling.
Well, I retired from my team. Yeah, because I have to start going into the office, and it's going to take me like 2 hours to get from the office to the bowling alley.
That's hard.
So
yeah, that makes sense. Maybe you need to start going to the Pacifica bowling.
Yeah.
Alley.
Or
a team in Pacifica.
In San Jose, or, there's
something not 2 hours away.
Yeah. There's other options. I'll figure it out. But then I'd have to not bowl with the people that I bowled with for 7 years, which would be sad. But,
that would.
But the team that won first place gave me one of their first place trophies. So that's my new trophy. That's like right here. Boop.
Ooh.
Is a first place trophy because my 10th frame gave them first place because we were playing the team that was against them for first place. And so because I clinched that last game, our team earned a point. It gave us like 4th place or something like that. But it put the second team out. And so this other team won first place. And so they were very thankful. And it was a team that I've been playing with for all 7 years. They've been in the league. Yes. And so we know each other, and they were all very psyched. And they were all watching me play because they were done. And it was very intense. And so, yeah, they gave me one of their first place trophies, which was very sweet.
So I can't really tell. Is that a guy fox mask in the
yes.
inside of the Rebel? That's amazingly beautifully meta. Perfect.
Yeah, Brian did that, obviously.
That's fantastic.
He also regularly puts on the Mandalorian mask. He'll just put it on.
Whoa! Let's just keep this conversation kid friendly.
No, he'll return to his schoolwork, just in his helmet at the computer.
He's like, I just need to wear this while I do my schoolwork.
And you know what? I can't blame him. It's great.
That's fantastic. And what is this book on like the little music stand. Is somebody?
it's the Princess Bride. It's an illustrated version of the Princess Bride.
Okay.
And we were looking for a bookshelf on Facebook Marketplace. And this contraption came up, a book stand, and we were like,
perfect.
Well, we have to buy that. So also Brian will periodically come in and change the page to another illustration. So if you go back through the YouTube pages, they're almost always different.
Yeah, I'm only just now
Yeah.
really focusing on the background.
He's a curator of sorts.
We'll find frames in past episodes and do a stop action of the book changing pages.
Yeah.
That's great. Mm-hmm. When did TWIS start on KDVS radio?
1999, 2000, something like that.
Yeah. Yep. Exactly. 10 years, Blair.
Mm-hmm.
I remember.
In the previous millennia.
I remember when you were just like this tall.
No.
How did
Incorrect.
Remember when she was babysitting Kai?
Yeah. Mm-hmm. Yeah.
No. Mm-hmm. She was
remember when I was wearing a cocktail dress to TWIS and giant hoops? I do remember that. Yes.
Yeah, you were
going to go clubbing.
Yeah. Was it clubbing? I thought she was going to go see the re-release of Beetlejuice.
How dare. Listen, I had a style. I have a different style now. But I enjoyed that style, and I look back on it and I have no regrets.
I am not judging in the negative at all.
Okay.
But
Beetlejuice is a great movie.
It was a great movie.
Yeah.
Beetlejuice?
Right? Yeah.
Don't
Have
say
you
it
not
again.
seen this? Oh! I almost did. I almost fell right into the trap.
Don't say it again.
Have you not seen this? Get out. Oh, man. No, I wasn't emo. I want to make that very clear. I was not goth. I was not emo. I just had like a very specific style.
You were a San Francisco girl who dressed all in black, but not goth because you don't want to be pigeonholed into that type of girl who dresses all in black. There's actually quite a myriad of things you can be all dressed in black growing up in San Francisco. Could be a Cure fan.
That works.
That would work.
Which is kind of goth, though, isn't it?
No, I was not goth. I was not goth at all.
No. Okay. Black lipstick?
No.
Okay, never.
You know it was red.
She had red lipstick. It was very red.
Yes.
Very red.
Very red. I like a bold lip. I still like a bold lip. I just don't do that anymore because of masks.
I tried.
Kind of pointless, huh?
I went through.
This has probably really hurt the lipstick industry, hasn't it?
Let me tell you, I recently... So, because I moved in the middle of the pandemic,
cares?
I lost all of my lipstick. I had no idea. Honestly, I had like probably 6 flavors, colors, whatever you want to call it, of lipstick that were my lipstick. And then they were all in a little pouch. And because I hadn't used it for 18 months, no idea. It moved. Somehow it's in this house. And I went to put on some lipstick and I was like, I guess I have to buy new lipstick. This is something I just have not thought about for 18 months. Yeah. It's pretty great. I only had to do eye makeup every day when I was working summer camp. Didn't have to worry about the rest of my face. It's great.
I tried a bold lip for a while, right around the time that Hedwig and the Angry Inch came out. And it was my goal to learn how to do that glitter red lipstick that they did. So my friend taught me how to do the glitter red lip. And so I had this really red lipstick, and I had a pot of really fine, awesome red glitter. Yeah. Make it fancy. Yeah. I have shiny lips all night long.
And then you left glitter on every cup that you used. And
No, I loved it. I'd go to a party, and I'd be like, I'm going to kiss everyone, because I'd leave glitter on everyone.
Oh my gosh.
And
It's
you
like
get
that
a lip on your
handshake
cheek.
test.
And you get a lip on your
Oh.
cheek.
It's like oh no, what was it? It was a buffet test at the start of the pandemic. Do you remember that?
Mm-hmm.
So they had one person at a buffet. They put like fluorescent bacteria on their hand or whatever, and then everybody served themselves at the buffet.
Oh.
And
Good night, Anthony.
there wasn't any real food. It was all fake food and stuff, right? It was like it was all miming. But at
Yeah,
the
I
end,
remember.
it was everywhere.
Was this a Mythbusters thing or?
No,
'Cause
I think
I saw
it was,
something where they did that on Mythbusters too, where they
Okay, maybe.
and then they had a party, and then they said, okay, one person in there had this fluorescence on their hands coming in, and this was their cold, right? And then at the end of the people just having a get together, which nobody knew that they were part of. They then put on the black lights, and you could see it all over everybody's faces and everywhere that they touched. This thing had transferred.
Yeah, I mean, this isn't the first one, but there were a bunch that happened right at the start of COVID because they wanted to explain why buffets were canceled and all this kind of stuff.
Yeah.
But yeah,
because
this
of
one was in
glitter.
Japan.
Buffets were canceled because of glitter. I think I remember what you were talking about, but yeah, stuff ended up everywhere.
All right.
Here.
I'm going to try and figure out how to manage allowed words, what words are allowed and not on the live chat. See if I can set that a little bit more firmly.
Oh,
Yeah,
do
well-
we have that power. Okay.
You wouldn't have thought you'd have to type these words into that, but that
Yeah.
sucks.
Yeah. And I would think it would be easier to find information on how to change them. I will keep looking. See what I
Yeah.
can do.
That was the other. That's right, Gord. The other little thread of that MythBuster was Kari being a
A germaphobe.
germaphobe and actually coming out pretty clean.
Yep. Germaphobes are paying attention and not getting your gross.
Oh, and they did one on double dipping potato chips, and it turned out it spread
not-
everywhere. Double dippers.
Yeah.
Double dippers and people who use
It's
hate
like
speech.
just
These are
one
people we
person
don't
doing
need
a
in
double dip
society.
and there's bacteria in your dip. Yeah. Oh, yes, Aaron Lore. Looking forward to the germ-laden world.
We're gonna dive in. I'm gonna lick all the doorknobs.
And then we're all going to be out sick.
Hey, who doesn't want a good old-fashioned cold again? Remember, you're just like, oh, I got a cold. And people are like, all right, well,
No,
Stay
I
over
don't
it.
want it.
Yeah, no, it's too
I don't want it either.
So before we go, just heads up again that next week I will be joining this show late.
Oh, that's right. Okay, so that's next week.
Yeah.
Got it.
Yeah. So if you could
How-
put the
how
animal
late?
corner at the end, that would be great.
Don't-
Okay.
Yeah, okay.
I may
go
be
ahead.
missing Wednesday the 18th.
Okay.
So it's like 2 weeks from now.
Okay. Are you going to give me your StreamYard login?
Yes.
Great. Can we
Do a
play with it
practice?
before? Yeah, you go
Yes.
because I've never done it. Okay, great.
Yes.
Okay, I love
Yeah,
it.
I was just thinking about it and going, we
Look
should probably talk about
who's
this.
trying to get Employee of the Month. First month that it's even available. Boy, you don't miss a beat, do you, Blair?
I want my name on that plaque. I want my picture up on the wall.
Oh, yeah. You're on
Oh
it.
my goodness. Don't double dip. Wear your masks. Be careful out there, folks. We hope that you're doing the best that you can. Hey, somebody's supposed to say goodnight to somebody right
Say
now.
goodnight, Justin.
Good night, Justin. Say good night, Blair.
Good night, Blair.
Good night, Kiki.
Good night, everyone. Thank you for joining us once again for this Zoomie episode of This Week in Science. We do hope you have a great week. Take care. See you next week. We will be here, coming back after I end the broadcast, and then we come back later next week. Okay, bye.